Okay, sorry about that. So let's go back. Finish up with 5A. Let's go to 5B real quick. Abnormal hemoglobin is identifying characteristics of sickle cell anemia. Explain the genetic basis of abnormal hemoglobin. Explain why the sickle cell allele is selected for in certain areas of the world. So if we're going to go through, remember hemoglobin is it's a protein. Um, so it's a protein that basically makes the blood red. It's technically it's I mean it's the red blood cell. I guess if you want to kind of take a look at it that way. So in the case of sickle cell anemia, the hemoglobin itself takes on a different shape, and it's not the very efficient kind of flattened disc that we have talked about in the past. It's more of obviously a sickle or kind of like a crescent shaped, which is obviously a big problem in some cases of not being able to transport oxygen very efficiently, getting caught up in the blood vessels, circulatory system, clotting, stuff like that. Explain why sickle cell alleles are selected for in certain areas of the world. Remember, it allows those individuals to become immune to or at least resistant to malaria. So the case, if they live in a part of the world where malaria is relatively is a high rate, therefore they will not die from malaria if they have the allele. However, if they have both alleles, it's almost too aggressive of a form that they're probably going to die from the sickle cell anemia itself. And so again, kind of the best case scenario there is that they have the heterozygous form of it, or at least they have one allele. Therefore, they're resistant to malaria, but they are also, in this case, um, also not. Sorry, a lot of stuff going on today. So they're also not going to die from the sickle cell anemia itself. All right. Let's go with thinking practice questions quickly. Number one, if the fine molecules are undergo a dehydration synthesis, remember water is given off. What molecule to result? So dehydration, we're bringing these two amino acids together. This part of the hydroxyl group right here is going to be released. That hydrogen is going to be released, forms water. Water is released. And you're going to see a bond between the carbon and the nitrogen here that would form that amino acid sequence. So you should be able to draw that resulting molecule. So circle the parts of the amino acid that would interact. Bond would form between those two right there. Identify which six main elements are found in each of the four macromolecules. Carbon, hydrogen, and oxygen you're going to find in every single one of these. Nitrogen, sulfur, phosphorus you'll find also in proteins, and then phosphorus you're going to find also in nucleic acids. So you've got that going on. Three, describe the relationship between substrate concentration and the reaction rate. So here we're seeing the increase in rate of reaction, right? And eventually that rate of reaction starts to plateau off as the substrate concentration goes up. So which basically means that substrates being produced, right? Well, one, we could take a look at all the active sites and the enzyme are being basically spoken for, taken care of, or simply there's just not enough of the initial substrates to be taken in by the enzymes. So eventually, what's going to happen is that rate of reaction is going to start to slow down there as we see. Four, DNA polymerase used in PCR. PCR is a technique where millions of copies of DNA can be made from one original copy. In this method, DNA molecules subjected to temperatures over 95 degrees to make the double-stranded DNA separate. Temperature is then lowered slightly to allow primers to anneal before the polymerase catalyzes reactions to incorporate the new nucleotide in the complementary strands. The cycle is then repeated over and over. Question A, predict why this bacterial polymerase is used instead of human polymerase. Remember, if it's human polymerase, what's going to happen after 95 degrees Celsius? That enzyme is going to become denatured. If it becomes denatured, then the catalyzation will not, or the reaction will not happen itself. So I would probably take the bacterial polymerase because bacteria has the ability to live in high acidic, high temperature environments, in this case, high temperature. Therefore, it would be able to function no matter what or I should say, if the temperature does get above 95 degrees Celsius. What would happen if you used a human polymerase in a series of PCR reactions? Again, I think you're just going back and answering question A, and that basically it would become denatured due to the increase of the high temperatures, and therefore the reaction wouldn't happen whatsoever. Sorry for the cutoff there. Make sure it doesn't happen again. But again, if you guys have questions on any of these, please ask. I will help you out with anything as far as these questions go. I don't think in the exam that you guys have in the next or whenever they decide it's going to be. I don't think you're going to have some that are so really specific as you see here. But again, just getting a basis for how to answer these questions, how to put them in order, what to write, little things like that. So yeah, if you have any other questions, please let me know. I am going to create one assignment and I'll put it up on Google Classroom for you. There's an FRQ dealing that I'm going to have you guys work on. I would like it due next Tuesday. And that's going to kind of just show me that you guys are at least following along and going through and doing what you guys need to be doing to prepare yourself for the exam. So I'll post that on Google Classroom here on Friday and give you guys a couple days to work on that and turn that into me in some way, shape, or form. But I'll put all that as far as in the directions. Again, good luck with the rest of the stuff. You guys need to get a hold of me, email me, get a chance. You need my number for text. Please feel free to contact me any way that you guys possibly can.